Hello, everybody. My name's Elizabeth Harvey, and Kateri Salt Gunderson and I are both the co-award chairs for the ASLO Awards Committee. One of the privileges of having this role is being able to announce the recipients of the ASLO Awards. But before I get into that, I do want to make sure to thank everyone who put in a nomination, wrote a nomination letter, or is part of one of our subcommittees that evaluates nominations. It's a lot of work, a lot of your volunteer time, and we really appreciate that. Nominate early and often, I say. Thanks, guys. The G. Evelyn Hutchinson Award intends to honor those who have made considerable contributions to increasing the knowledge in areas of limnology and oceanography. This year's awardee has made numerous groundbreaking contributions to understanding the complex interactions between cyanobacteria and the viruses that infect them, leading to the development of new research directions for the entire field. As one of her colleagues stated, her entire career has been universally creative, rigorous, and path-breaking. Beyond the lab, the awardee has also been active within our profession through society and editorial service, as well as undergraduate and graduate student mentorship. So if you would all please join me in congratulating this year's award winner, Dr. Debbie Lindell.
Thank you very much, Liz, and thank you everyone for coming. It is really an honor to be standing here before you, and I would like to begin my presentation by thanking many people. First of all, I'd like to thank ASLO and the Awards Committee, my nominator and letter writers, for this recognition and award that they have bestowed upon myself and my team. It is truly humbling to receive an award holding the name of G. Evelyn Hutchinson, an eminent ecologist of great standing. It is also extremely humbling when one looks at the list of people who have received this award, many of whom I know and have a great deal of respect for. So thank you very much. I'd now like to move to thanking my mentors, and in doing so also provide with you a little bit of an indication of my pathway, my scientific pathway, and also give you one takeaway that I've taken from each of my mentors and tried to incorporate in my own mentoring of my own students. First of all, Amatsi Ganin, who is here at this meeting. I was an undergraduate who finished a biology degree and wanted to know whether biology was something that I wanted to work in. And I applied for a position in his lab as a research assistant, got that position, and you could say that that's actually the reason why I'm in oceanography today. I worked with Amatsia on reef fish planktivory, and one of the main things that I took from Amatsia, there are many, but one of the main things that I took from him is leading by example. I then went on to do a master's with Anton Post, who introduced me to the world of cyanobacteria, and studied the succession of phytoplankton in the Gulf of Aqaba over the seasonal cycle. And I also went on to do my PhD together with Anton and Aitana Padan, a molecular biologist who helped us bring molecular biology into our research to try to understand the nitrogen status of Synechococcus, to understand whether the decline of Synechococcus over time in the Red Sea was due to nitrogen limitation. And I can say that the combination of ecology that I got from Anton and molecular biology that I got from Aitana, and the combination of that is a thread that is stayed throughout my research since then. One of the main things that I took from Anton is understanding the importance of collaborations. And one of the main things that I took from Aitana was understanding that quality over quantity is important in science. I then moved to MIT to work with Penny Chisholm as a postdoc, and it is there that I began working with viruses, and viruses of Prochlorococcus. And one of the major things that I took home from Penny is to look at the big picture and to put my research into a larger context. I have been really, really lucky in my mentors, and I'm very grateful for everything that they have given to me over the years, and I thank them greatly. And now to my team, past and present. Clearly, a prize that is given to me, or an award that is given to me, is actually not just for me, it's for my entire team. They are the ones who've done the research, and it has been amazingly stimulating to work with a great group of research associates, students, and postdocs. And it is probably one of the most enjoyable parts of my career to help mentor them and to guide them to find their way and to reach their potential. And it's a great source of joy for me to advise all of these amazing students. I don't have time to go through names. I will be presenting work that is by these people, and their faces and names will be on those particular slides. I've also been extremely lucky to work with a great group of collaborators, both in Israel, in my institute, and elsewhere, in Europe and in the US, and many PIs and students as part of the SCOPE program. And I'm also now involved in an ocean margins initiative and gaining new collaborators. The work, I'm very grateful to the work that I've done together with these people, that we've forged different directions and reached different levels of science than what we could have previously. All of the work that I'm going to be presenting to you today is collaborations with a number of these people. I won't have time to point out all of the people, but just know that all of the work that I'm presenting is collaborative. I'd like to also thank the funding agencies who've provided me with the funds to be able to carry out a lot of this research. I'd like to point out in particular the Israel Science Foundation, the European Research Council, and the Simons Foundation, who funded me for 10 years or more at high levels and enabled me to carry out the research and take it in the directions in which I've wanted to. And lastly, but certainly not leastly, is my family. My partner, drawer angel and oceanographer at Haifa University, whose boundless support and belief in me and gender equality belief that is borne out in actions has really been amazing, and I probably wouldn't be standing here today without his support, so thank you very much. Our two children, Gilad, our son, and Danielle, our daughter, are great sources of enjoyment and pride, and now as young adults also support for me, and their joy in my successes is very, very heartwarming. And I'd also like to point out their partners, Shai and Margot, who's very enjoyable to spend time with as well. And lastly, my friends and teammates who keep me sane, who keep me grounded, and somewhat fit. Okay, so now I'd like to transition into the science part of this talk, where I'm going to speak to you about host virus interactions in cyanobacteria. And I hope to be able to impart to you the impact of interactions at a single cell level and how they can have important ecosystem effects. Okay, so the organisms that we work with are the unicellular picocyanobacteria, Synechococcus and Prochlorococcus, which all of you are well aware contribute significantly to global primary production. They have different distribution patterns in the oceans. They also have different pigment contents, which is important for us in oceanography and enables us to differentiate between them using a flow cytometer. And they also, very importantly for today's talk, co-occur with many viruses. So viruses are basically protein coats in which exists genetic material, often DNA. When a virus infects a cell, it has to first be able to recognize an extracellular molecule on the surface of that cell. Once it recognizes that cell, it can attach to the extracellular surface, and then it can insert its genetic material into the cell. This is essential because viruses cannot replicate outside cells. They are obligatory parasites, so they must recognize a cell, attach to it, and introduce its genetic material into it, replicate that genetic material, express that genetic material to produce the proteins that make up the virus, and then once the viruses are made, they need to make their way outside of the cell. And to do so, many viruses, and the viruses that I'll be talking about today, they lyse the cell. They kill the cell in order to be released from the cell, and then to go out into the water column and find a new host. In doing so, they break open the cell. So here you can see an electron micrograph of a virus attached to a Synechococcus cell. You can see viruses being released from the cell 9 hours after infection, and all the glob that comes out of them, and the shell of a cell that is left after that infection. Okay, so the overarching goal of the research in my lab is to understand how viruses impact the Picocyanobacteria and in turn the ecosystem. And specifically for today's talk, I'm going to present 4 vignettes that I hope provide you with an understanding of the overview of the different ways in which viruses impact marine ecosystems. I'll talk to you about the effect of viruses on population dynamics and distribution of the cyanobacteria, how viral lysis can impact ocean biogeochemistry, how resistance to infection is a means for host-virus coexistence in the oceans. And also, I'll end by telling you how viruses have stolen genes from their hosts, incorporated into their own genomes, and now use them against their hosts to produce themselves. Okay, so to begin with, to be able to understand how viruses impact their hosts, we need to be able to quantify their viruses, and perhaps even more so, we need to be able to quantify the extent to which they are infecting their hosts. Surprisingly, to do this in a host-specific manner or in a taxon-specific manner is really not trivial and was not possible for many years. We borrowed a method called the Polleny method from a different field entirely and brought it into viral ecology. And what we basically do, what this Polleny method does, is uses PCR. We all know what PCR is for detecting viruses because of COVID. But basically what it enables us to do is take a single molecule of the viral DNA and amplify it up to a high enough level that we can actually detect it. But for viral ecology, because of the massive diversity that exists in viruses, we can't do a normal PCR reaction in the tube because there will be no real quantification. The error levels are 100-folds or more. So what we do is we do a solid phase PCR, which basically means that we spatially separate the viruses inside of a gel, and then there's no competition for the components of the assay, and in this way we're able to quantify with an error level of 2-fold. So we can do ecology here. So this is how we quantify the different families of viruses that infect cyanobacteria. We use their DNA as a barcode, much like you do when you go to the supermarket and scan your produce. Okay, so we can now quantify viruses that infect cyanobacteria, but what's even more important is being able to quantify the extent to which they infect the cyanobacteria. And to do this, we first use the different pigment complements to separate Synechococcus and Prochlorococcus using flow cytometry. And then we embed the cells, single cells, into the gel. And then we do the PCR, the solid phase PCR, but we look for viral DNA that is inside those cells. And then every cell that has viral DNA inside of it is infected. And we can interrogate thousands of cells at the one time and get robust values of infection. Okay, so we now use this method to assess viral infection and viral abundances in different regions of the ocean. And the work that I'm going to present to you today is work done as part of the Gradients Scope program, where we traveled from Hawaii due north in 3 different cruises from the subtropical gyre through a transition zone towards the subpolar gyre. And there's gradients in all sorts of environmental parameters, and we looked at the abundance of viruses, the different types of viral families that infect cyanobacteria. And what we found was that we have quite high levels of viruses, 3 to 5-fold more than the cyanobacteria in the subtropical gyre. And we found that there are 2 families that are most important: in yellow, T4-like cyanophages, and in blue, the T7-like cyanophages. But that when we traverse north, we see that there's an increase in both of these families, but whereas the T4-like cyanophages were most abundant in the subtropics, it's the T7-like cyanophages that are most abundant in the region between the 2 gyres. So we have differences in community composition, and we have a change in the abundance of viruses with environmental conditions as we go north. When we now take these 3 cruises and plot them, instead of by latitude, by temperature, we see that we can somewhat normalize for the differences in the transects and see that this hot spot of higher numbers of viruses occurs at a consistent temperature range. What we can also see is that in June 2017, there was approximately 500,000 more viruses per milliliter of seawater than in the other 2 cruises. We can now use this way of presenting it to compare the abundance of the viruses to the abundance of the cyanobacteria that they infect. And what we can see is that in the year where we had 500,000 more viruses per milliliter, we have a crash in Prochlorococcus that is anti-correlated to that increase in viral abundance, suggesting perhaps that viruses are causing this crash at a higher temperature than normally happens or that happened in the other 2 years. When we now look at infection by using the Polanyi method and detecting viral DNA inside cells, in the June 2017 cruise, we see that we had infection levels that went from less than 2% in the subtropical gyre to up to about 10% in the surface waters. And so we have a massive level of infection, much higher than in the other 2 cruises, even though those 2 increased in the region between the 2 gyres. If we now estimate the degree of mortality prior to the division of Prochlorococcus at this temperature, we get that between 30 to 50% of Prochlorococcus could be killed by viruses prior to their ability to replicate, to reproduce. So we have here clear evidence that cyanophages, viruses that infect cyanobacteria, can dramatically impact the biogeography of cyanobacteria. All of this work was done at the surface waters, and when we now look at depth profiles, what we can see is that viral abundance and viral infection increases with depth, more so between the gyres than in the subtropics. And we see that we get infection levels that were beyond our wildest dreams during the June 2017 cruise, reaching up to levels of beyond 50%. So viruses can have a massive impact on cyanobacteria at some times and less so at others. Okay, so now I'd like to move to the second vignette of looking how viruses impact the fate of organic matter. The role of viruses in stimulating the microbial loop has been discussed and known for many years, back to Jed Furman's work, Curtis Suttle's work, Marcus Weinbauer, Steve Wilhelm, and many others. Basically, what we know that happens with lysis of cells is that all of the organic matter that's inside those cells is released into the water column and can be taken up by other phytoplankton or bacteria and stimulate the microbial loop. over the past decade, it has now become clear that viruses can also cause stickiness during infection and after lysis that can lead to particle aggregation and potentially also export out of the photic zone. If we now have a look at the transects that I showed you, looking at the potential for viruses to provide carbon to the microbial community, during the June 2017 cruise, we can see in between the gyres that viruses could contribute between 20 to 30% of the microbial carbon demand, whereas in the subtropical gyre, this is only at around 4 to 5%. So they can contribute dramatically to feeding the microbial community, but this is different in different regions and in different times. If we now want to have a look at a little bit more detail how viruses can stimulate both the microbial community as well as particle aggregation, and to see whether Prochlorococcus, which is a small, neutrally buoyant cyanobacterium that is not really considered to be something that's going to be causing aggregation or particle formation and export. We carried out a joint collaborative experiment in the lab with 10 different labs as part of the SCOPE program, where we have a single Prochlorococcus, a single virus, a single grazer, and co-occurring heterotrophic bacteria. And when we look at the abundance over a 2-day period to have a look at the impact of the grazer and the virus and the combined grazer and virus, we see that Prochlorococcus grows nicely without any mortality agent. In blue, we see the decline of Prochlorococcus with low virus abundances, and in dark blue, the decline in the presence of high levels of viruses. And we can see the concurrent increase in heterotrophic bacteria as these cells are lysed, with the organic matter being utilized by these viruses. We don't see that in the presence of the grazers, even when viruses are around, because the viruses are feeding, the grazers are feeding upon these bacteria, and we've got rapid turnover of the microbial community. When we now have a look at aggregation, what we can see is that there is a small degree of aggregation in the control. And we're talking here about aggregation that we can see in flow cytometry. So these are fluorescent aggregates that still have chlorophyll from Prochlorococcus in a window of less than 10 microns in size. And what we can see is that in the presence of the grazer in orange, we have some aggregation. In the presence of the virus in blue, we have some aggregation from about 12 hours onwards. But when we have both grazer and virus, we have massive aggregation that occurs, and this is reproducible in 3 different replicates. And when we then sort these aggregates and look at them under a scanning electron microscope, we can see their beautiful form. We can see grazer scales. We can see what looks like damaged Prochlorococcus cells. We can see bacteria that are colonizing these aggregates. And so certainly the interaction between viruses and grazers is impacting aggregation. We don't know whether these are stay buoyant or are heavy and being exported, but they are certainly substrates for bacterial colonization and remineralization on these particles. Okay, so now I'd like to segue into the third vignette. I'd like to remind you that I spoke about very low infection of cyanobacteria in the subtropical gyre, even though there is 3 to 5-fold more viruses than Prochlorococcus in that region. So there could be many reasons for this, and one of the reasons that we think is likely to be of great importance is resistance. Perhaps many of the Prochlorococcus in that region, are resistant to infection. So this has been a topic that we've been working on in my lab for quite some time. And just to remind you that a sensitive cell is infected by a virus, goes through all of the stages of infection and lyses the cell at the end. Viruses are produced and the cell dies. When there is resistance, the infection cycle is halted at some stage during the process. It can be at the very first stage, where there's a change in the extracellular molecule that is recognized by the virus, and then the virus can't even attach and enter its genetic material into the cell. Or it can be later on in the process. Either way, no viruses are produced and the cell survives. Okay, so to look at resistance as a potential means for coexistence, we have carried out we've used a number of different approaches, and I'll just present one of these approaches to you today, where we use selection for cells that are resistant. And what we do here is we take cells that are sensitive, Prochlorococcus and Synechococcus cells that are sensitive to cyanobacteria, we allow them to replicate. And during replication, there are normal errors that happen in DNA replication, and this causes the diversity of genetic material in the population. And then we challenge the sensitive population overall with different viruses, and all of the sensitive cells are killed off, and the only ones that remain in the presence of the virus are those that have developed resistance to that cell, to that virus. Sorry. Then we can go and characterize them both at the genotype level and the phenotype level, and have a look at what this resistance has caused or is manifested. And so what we found is that when we use this approach, that many of the mutations that conferred resistance were in cell surface molecules, preventing the virus from recognizing, attaching to the cell, and introducing its genetic material into the cell to be able to replicate. And this, you can see by the various different types of mutations that occur, that we're getting a diversification of these cell surface genes at the genetic level of a particular gene, but also a diversification of cell surface type molecules in these populations of cyanobacteria. Well, one might think, well, if you've got resistance, then why aren't all cells resistant? Because then you survive. And part of the answer to that is that there's a trade-off, there's a cost to being resistant. And the most well-known cost is that of growth rate. And when we looked at our resistant strains, we saw the ones here that are shown in green. These indeed have a lower growth rate than the sensitive strain that's shown in blue. But what we also notice is all of these red strains in red that had no growth rate costs, at least under the conditions that we tested. But when we had closer look, what we noticed was that this resistance to one virus caused a greater sensitivity to other viruses. And we can think about it in terms of, if you have a cell surface molecule that viruses recognize, and you change that in some way, and that causes resistance to one virus, perhaps the same change can actually enhance the ability to attach for other viruses. And this is exactly what happened. We get enhanced attachment and enhanced decline of the population. So resistance to one virus can lead to greater susceptibility to other viruses. So it's quite a complex system. And what are the ecological implications of these findings? So if you have a population of cells, of Prochlorococcus cells, that have different surface molecules on them, and a virus that I've depicted here needs the orange extracellular molecule, it can only attach to cells that have that orange molecule. The other cells that have different molecules are not recognized by this virus. This virus then goes and infects and kills off those that it can, and leaves those that have diverse cell molecules on its cell surface. So what we basically got is a diverse set of subpopulations of Picocyanobacteria that prevents the decimation of the entire population and increases population diversity and community resilience. And now we have coexistence where some cells are susceptible to viruses and can be infected, so we get the viruses reproducing, and other cells are resistant to viruses and maintained. And so we have host-virus coexistence over the long term. Okay, I'd now like to move to my last vignette, looking at the effects of viruses on host photosynthesis through these viruses making use of the cell's own genes that they have stolen from them and incorporated into their genome. So about 25 years ago, we as a community started doing genomics and were able to sequence cyanobacteria, all sorts of different microbes, and viruses that infect them. And we discovered an enormous degree of diversity in these microbes, as phytoplankton and the viruses, with many genes of unknown function, particularly in viruses that infect all sorts of organisms. But perhaps what was more surprising than the level of unknown genes that we had was the finding that there were some genes that we knew very well. In the viruses that infect cyanobacteria, we found photosynthesis genes. And viruses certainly cannot carry out photosynthesis. They don't have the machinery, the rest of the machinery to do it. They just have a gene here or there. This was first discovered by Nick Mann in Synechococcus viruses, and later by myself and Matt Sullivan when we were in Penny Chisholm's lab in Prochlorococcus viruses. And over time it became clear that about 80% of the viruses that infect cyanobacteria have photosynthesis genes. But not only that they don't only carry photosynthesis genes, they also carry carbon metabolism genes, nucleotide synthesis genes, nutrient uptake genes, et cetera, and that this isn't only something that occurs in viruses that infect cyanobacteria, but in viruses that infect many different organisms in the oceans. And this led to many hypotheses as to what these genes are doing. But when you look at the sequence and you're left at the level of genomics, all we can do is raise hypotheses and not really test them. So this led to a great motivating force in my lab to develop a genetic system that would enable us to test hypotheses as to what these genes are doing. And what I mean by developing a genetic system is basically a way in which we can take a normal virus, a wild-type virus, and compare its activity to a virus that has everything else except for the one gene of interest we're looking at. And then we can assess the benefit of a single gene to the virus and the effect of this gene on the host. And so in using this system, I'm going to show you one story of a gene called NBLA that is a cyanobacterial gene that viruses have taken and put into their own genome. First of all, I'd like to start off by giving a little bit of an introduction into the light harvesting complex of most cyanobacteria, of Synechococcus, but not Prochlorococcus, and many other cyanobacteria. This is the phycobilizome. This light harvesting complex attaches pigments to it, which are used to collect the sunlight and transfer that energy from the sunlight to the photosynthetic apparatus for chemical energy production. These light harvesting complexes are large protein-containing complexes that can hold up to 50% of the cell's proteins, and they contain a massive amount of resources. Jackie Collier discovered a gene called NBLA in cyanobacteria that is activated during nutrient limitation in freshwater cyanobacteria and goes about to direct the degradation of Synechococcus's light harvesting complex, causing loss of pigmentation or bleaching, and the release of amino acids that the cell then uses to survive during the period of nutrient deprivation. So the question that we were asking is, what is this gene doing in viruses? So what we did here is we genetically modified the virus, we engineered it to lack the NBLA gene and compared it to the normal virus that has the gene, and that's the only difference between them. And when we do that, we can see that the infection cycle of the normal virus with the gene is threefold more rapid than the virus lacking the gene, and that more viruses are produced. When we look a little bit more deeply into the molecular level, we see that the normal virus with the gene can produce many more of the proteins it needs to make its particle. So we're hypothesizing that the amino acids that are released from degrading the phycobilisome are not being used for cell survival, but the virus is in fact using it to degrade the host's photosynthetic light harvesting capacity to produce new viruses. When we have a look at the impact on the host, we can see that the virus with the gene has loss of pigmentation. The virus lacking the gene has much less loss of pigmentation. And this leads to a decline in photosynthetic efficiency much more rapidly in the virus that contains the gene. So if we now want to have a look at what the impact of a single gene might be on cyanobacterial photosynthesis in the oceans, we take into consideration the distribution of the gene, the abundance of the gene in a family of viruses that contains it. We look at the extent of infection from the Polanyi method that I presented to you in the beginning, and we take into consideration the reduction in photosynthesis. And when we do all that, we estimate that this particular gene in viruses could be reducing global cyanobacterial photosynthesis from anything from 1 to 5%. Okay, so I'd like to finish up now by trying to bring it all together and tell you that there are many ways in which viruses can impact the ecosystem. They can impact the population size of their host. They can impact the diversity of their hosts. They can impact the metabolic output, and they can impact carbon cycling. And all of this begins at the intracellular battle between a single virus and a single cell. If the virus is successful and produces progeny, it will kill the cell. It will lead to recycling of organic matter that is utilized by other community members, and a substrate for aggregation of the cells. If it's not successful, we will have resistant types that will continue to grow. And in fact, what we have is a combination of all of these. And I'd like to just leave you with what I consider to be one of the major challenges in oceanography now, and that's to incorporate viruses into ocean models in a meaningful manner through utilization and obtaining empirical data, not just for cyanobacteria, but for many phytoplankton and microorganisms, so that we can constrain such models and they can be informative for us for understanding our oceans. And I'd just like to thank everybody again and thank you for your attention.